Thank you for joining us today. Welcome to our bi-monthly webinar series. We hold this out of EPA's Office of Research and Development, which is part of the Safe and Sustainable Water Resources Research Program under the EPA's National Water Research Program. Today, Teresa, Dave, and Ning will be presenting on their mayflies and mussel research. But before we get started, I would like to discuss a few housekeeping items. If you have any technical issues, please send an email that can be found on the screen or enter into the chat box any issues that you are having. There is also a handout section near the chat box on the navigation bar, which will have today's slides if you need a copy to download. We will try to get through a short Q&A session at the end, but cannot guarantee that we will get through all the questions since we have a lot of material to cover today. But please feel free to enter any questions you have in the chat box. We also do have the contact information on the slides if you'd like to follow up with a presenter if we do not get through to your questions.
Okay, great, Nikki. This is Michelle, just a couple things. So during the Q&A session, we would actually like if you would type those questions in the questions box. There is a chat box for general comments, but the questions box is where we monitor questions or any issues that you're having during the webinar. Also, you can see here we have, just so you know, we do have links to our program if you want more information on this research. Also, if you missed the poll, if you're just joining us, we did have a poll we like for our outreach efforts to keep track of how many people are actually viewing this webinar. So if you have people in the room with you who are not logged in and are viewing this webinar with you, if you could please put the number of people in the room with you in the questions box, we would really appreciate that. And also, because we do give out certificates, if you'd like one for attending this webinar, if the people in the room with you are interested in certificates, you can also type their names in there, and we'll make sure they also get a certificate.
Okay, thanks, Michelle. And here's this disclaimer for you guys to read. So today's webinar is in partnership with the U.S. Geological Survey and the Illinois Natural History Survey. To further protect aquatic life, there is a need to provide additional standardized test species for freshwater environments for chronic exposures. This webinar will highlight the progress of the mayfly and mussel research. So Teresa Norbert-King will be giving an overview of this presentation today and will be introducing Dr. Su and Dr. Wang. She is an aquatic biologist with our Office of Research and Development, and I will now turn it over to Teresa.
Hello, everyone. Show my screen, right?
You can go ahead, Teresa.
There we go. Okay. Just a second. There we are. I just wanted to acknowledge several of the people who have been working on this, which include the people Jeff Stevens, James Coons, Doug Hardesty, Amy Dickinson, Ed Hammer, Candace Bauer, and Chris Barnhart, and all of the other people that participated in the mussel interlaboratory study that Ning will talk about in the future. I wanted to give a moment for everybody who is on the call who may not be as familiar with the program of the whole effluent toxicity testing, and also to talk about that this was initiated with a regional applied research effort through Region 5 with Ed Hammer and Candace Bauer and Rob Pepin. And so I just want to give a few slides to talk about background and species that we're using. So I just talk about that we're using effluent testing to assess adverse effects of toxicity in an effluent to a population of lab organisms, and we're assessing the combined effects of potential contaminants in an effluent. It's an important component of EPA's integrated approach to protect the surface waters from pollutants, and it's typically included in the NPDES program. In the agency's technical support document for water quality-based toxics control, or is often referred to as the TSC, EPA recommends that for acute testing, which is pretty much lethality testing, that testing is done with 2 U.S. EPA-approved wet test species that are representing different trophic levels, such as in a fish and an invertebrate. And then for chronic or the sublethal effects, such as growth, survival, reproduction, we're looking at saying EPA should test at least 3 U.S. EPA-approved wet tests, including a fish, an invertebrate, a plant, or an algae. And then under both the acute and the chronic wet testing, each species should be assessed against a permitted effluent at least 3 times to determine the most sensitive. species for determining whether there's an excursion of a state water quality standard or for monitoring to determine the compliance with the NPDES wet permit limits. For toxicity testing, most people are a lot more familiar with talking about chemistry and the analytical detection, but in this case, we're actually using the organism as an analytical detector because in these samples, we don't have a really good idea what the toxicant is. So the animals are telling us this in the evaluation. Our toxicity test can be affected by a number of variables that are listed here, among others, as even the types of samples you receive and the frequency of the types of samples. So those are a lot of parameters that go into how a toxicity test is run. So while we say the test conditions are standardized, the method still will allow a range of acceptable conditions. So EPA has 3 manuals that are first developed for the acute and short-term toxicity methods. These were then promulgated in 2002, which means that certain methods are listed in the Code of Federal Regulations, just like many of the analytical analyses are. But because I think looking at manuals is really too much of a flat screen, I wanted to talk about what the species are so that you can actually see how the methods we're going to talk about with mayflies and mussels fit in. So this screen sets up to have the acute and the short-term chronic test methods, where the top of the screen are the species that we use for acute 48 or 96-hour tests, and the bottom are for the short-term chronic tests, which are, some of them are the same species and then the top. And then you can see that we don't have species for mimicking both acute and chronic for the freshwater. For the marine and methods that were published at the same time, we have 2 fish and a mysid, and we also have a sea urchin. So we have a few more species that we're getting at. If we talk about what we have for the West Coast marine and estuarine short-term methods, we have 1 more fish, and we have a lot more invertebrates, and we have a plant. So we have a few of these species that are being used, but we really felt like we needed to look at other species such as the mussels and the mayflies, which have been shown to be sensitive when tested with several metals and major ions. And then they've been used successfully in toxicity testing to support water quality criteria document development. And we're talking about the mayfly and the fat mucket. And Dave and Ning will both be giving more details about the methods that we've been working on to develop. So this RARE project was done with the goal of assessing their utility in an effluent test for both the short-term 96-hour test and then a 7-day test. We looked at longer terms, but we really wanted to pair them up with the standard short-term wet methods that already exist. So I just want to talk about why did we pick these certain mayflies. The mayfly is a parthenogenic species. It's found in a wide distribution and in many of our EPA regions, and it's been demonstrated to be particularly sensitive to major ions and chloride and sulfate that are the components of water conductivity and many metals. And so with the fat mucket, you see the distribution is broad throughout the U.S. and Canada, and it's been demonstrated to be among the most sensitive of the various species to several of the contaminants such as listed there. So the idea would be that we, for the invertebrate testing on the table below, we have what we might have for the acute mortality test. We would add a mayfly and a mussel. And then if we were to do the short-term chronic estimate test, I would see then we would also have a mayfly and a mussel. And this would allow us to have some taxonomic diversity and choices for an appropriate exposure. And with that, I think I am going to let Dave start to do his presentation. I also want to mention that for anybody who joined after the beginning, if you would please go to the question field, and if you have more than 1 person at your location for doing the presentation, that you would list the number of people in that question box for how many are there. And Dave is a researcher at the Illinois Natural History Survey. I think you could have read that. And I look forward to your presentation, Dave.
Thanks. Right. So as Teresa said, there's a need to have a sensitive insect species. Insects are aquatic insects are among the most diverse in stream ecosystems, and most important in terms of biomass in stream ecosystems in North America. And as Teresa alluded to, they're quite sensitive to metals and major ions, and also things like neonicotinoid pesticides. So we're working on developing methods for this species. And this is the species we're working with here. This is Neocloeon triangulifer. And if we, going back, there's not enough time to go through a full detailing of the history of the use of this organism, but going back to the 90s, Bern Sweeney and David Funk at Stroud Water Research Center developed a culturing and toxicity testing method for this mayfly. And as Teresa mentioned, it's useful for laboratory testing because it's parthenogenetic, meaning it can reproduce asexually. But the folks at Stroud developed a method for culturing and toxicity testing using naturally colonized periphyton plates to feed the organisms. Then in 2015, pardon?
Dave, I am so sorry to interrupt you, but we are seeing your presenter mode. Could you switch that for us?
Is that good?
Yes. Fantastic. Thank you.
I apologize for that. So as I was saying, so in 2015, Paul Weaver and Jim Lazorchek and the group there at US EPA ORD in Cincinnati then developed a method of culturing a diatom biofilm diet in the laboratory for this species, which is an important step moving towards standardization. We then adopted that method and built on it and continued to work to develop a method for conducting full-life chronic toxicity tests with this species using reconstituted waters with known composition and laboratory cultured diatom biofilm diet. And we published those results in a paper by myself and Amy Dickinson in 2015. If I can go back once, I apologize. So for this effluent testing project, our objectives were to try to move forward with development and improvement of methods for this species using these mayflies. And we had two main things that we wanted to look at. And the first bullet there is we wanted to identify optimum organism age at the start of the test with the goal of finding the best combination of maximum sensitivity and minimum test duration. And then secondly, we wanted to investigate influences of various aspects of diatom culture technique and on food quality and therefore mayfly growth. So what makes the food better with the assumption that improved control growth during toxicity tests will result in a more representative response to effluents. And this is similar to a process we went through with Hyalella azteca, the amphipod, a few years ago. Some previous work had been done comparing different ages. Jim Lazorchek's group gave a presentation at a symposium put on by Ontario Ministry of Environment, where they compared survival and growth, which they measured as length, of 1-, 3-, and 5-day-old larvae that were exposed to sodium chloride. And they observed significantly lower survival in terms of an LC50, medium lethal concentration, in tests with the 1-day-old as compared to the 3- and 5-day-old larvae. They didn't observe differences in IC25 for the growth, which again measured as length among the different age larvae. We like to use dry weight as our growth endpoint because it's more sensitive in the sense that dry weight of the organism is proportional approximately to the length cubed. So the weight changes in 3 directions, whereas length only changes in 1. So a given change in length results in a much larger change in dry weight. The difficulty is that it's hard to accurately measure a 7-day-old mayfly. So if you start with a less than 24-hour-old mayfly, at 7 days it's hard to get an accurate weight. But what we've done is we've made a relationship and plotted here length versus dry weight for a range of organisms, and developed a method of calculating dry weight based on body length. And it's a pretty tight distribution. And these are data from both our lab at Illinois Natural History Survey and from Jim Lazorchek's lab at Cincinnati, at US EPA ORD in Cincinnati. And so that's what we use as our endpoint. So we use weight instead of length. And the experiments that we wanted to conduct in this part of the project was to, first of all, compare sensitivity of less than 24-hour-old versus 7-day-old mayflies in 7-day tests. The reason that we chose the 7-day-olds to compare to the less than 24-hour-old mayflies was because, again, Jim Lazorchek's lab had done some intermediate ages 3 and 5 days, but if you start with a 7-day-old organism and test it for 7 days, by the time the mayflies are 14 days old, you can actually get direct dry weight measurements that are quite accurate. So if the sensitivity was all the same, getting a direct dry weight measurement might be useful. And then we also wanted to compare the sensitivity of 7-day tests to a longer term, like a 14-day test, and a full life cycle test. So looking at how the duration of the test affects sensitivity. The test conditions that we had for these 7 and 14-day tests, we used a temperature of 25 degrees Celsius, standard photo period, and light intensity, different size test chambers and solution volumes depending on whether we used less than 24-hour-old or 7-day-old organisms. For our dilution water, we used reconstituted water that was developed to mimic natural surface waters that was developed at the EPA lab in Duluth. There's no substrate except for food in these tests. And again, we started with less than 24-hour-old and 7-day organisms. And in many ways, this test, the way we set this up was to kind of mimic the Ceriodaphnia dubia test, in which we had 10 test chambers per treatment and 1 organism per test chamber. We fed the mayflies a scraping of a live diatom biofilm because we found that live diatom biofilms, the animals grow much better than at least in our lab research, than say, for example, a refrigerated diatom suspension. We renewed the water 100% replacement every day, which was something we were wondering how the organisms would do in that much handling. Again, we did 7-day tests and 14-day tests, and then looked at endpoints like survival, dry weight, and biomass. So to look at some results, this is just 3 different tests where we had, we started, the green bars were where we started with less than 24-hour-old organisms. The yellow bars are where we started with 7-day-old organisms. Both of these are 7-day tests, and it's just 3 iterations of the test. And so the values shown on the y-axis, these are the most sensitive endpoint, the EC20, and half of the test it was dry weight, and half of it was biomass, which was the total dry weight of all the organisms alive in the test. So it was never survival. But as you can see, there's a distinct and significant difference between the chronic values between the test started with less than 7-day-old organisms and those started with 7 days. So about a 4-fold difference on average overall. So quite a bit more sensitivity with the younger organisms. Compare that to longer duration tests. I've added a gray bar and a blue bar next to test number 1. And the gray bar is a 14-day test that was started with less than 24-hour-old organisms. And then the blue bar is a previously published test where we did a full life cycle test where we started with less than 24 hours old and went all the way to emergence as an adult. And that was the most sensitive endpoint there. So as you can see, comparing the 7-day test with less than 7 and looking at dry weight or biomass and comparing to longer duration tests, you get pretty similar, if not better, sensitivity compared to the longer term tests that we had. So that's quite encouraging. It looks like we should go with the less than 24-hour organisms and do 7-day tests. So the other part of this study, and it's a part that's still in progress, was to investigate influence of various aspects of diatom culture technique on food quality and how that affects the mayfly growth. Again, with the assumption that if they grow better, their results in these toxicity tests will be more representative of their responses to effluents. So when we started looking at this, we noticed that when we use this reconstituted water, where we take a deionized water and add salts back to it to get it to a composition that we want, we had a variety of weights and lengths. So I'm just plotting length and then the calculated dry weight of those lengths after 14 days and just controls with no contaminants included. And you see that they ranged quite a bit, quite widely. And when you compare those to similar test controls that various tests that we conducted in natural waters, for example, collected from streams in Illinois and in Minnesota for other projects, you see that on average, when these organisms are grown in a natural water, they grow much better. Their lengths and weights are consistently higher, and the variability is actually less as well. So we got to thinking that we need to do, it's possible that there's something about these natural waters that's contributing to the quality of the food that these mayflies are eating that we may need to try to mimic in a reconstituted water. So we decided to do an experiment with our diatom cultures, wherein we looked at nutrient concentrations over the course of 7 days. So we sampled the nutrients in nitrogen, phosphorus, and silicon in the water on days 1, 3, 5, and 7 after the initial initiation of the culture. And the y-axis is just the percent of the initial concentration of the various nutrients there. And you can see while nitrogen remains relatively stable, it drops some over the course of 7 days. Phosphorus and silicon drop to about half over that period of time. So these diatoms are really taking up a lot of silicon and phosphorus, and that might and suggesting that these two nutrients are limiting to these species, and if they are reduced in time in the culture, the quality of the diatom biofilms might be decreasing. So we did some experiments with mayflies where we created an amended lab water, where we took our reconstituted water with a hardness of about 100, and we added nitrogen and silicon and phosphorus, and then also some B vitamins and biotin. And then we just did some comparisons of 14-day growth with no contaminants in this case. And we did these tests 3 times, and then the third time we tested treatments with 1 organism per beaker, and then treatments with 3 organisms per beaker. That's the triples and singles in test number 3. But what you see, so on the left, the left bars are the standard reconstituted water with no nutrient additions. On the right is the amended with the nutrient additions. And as you can see, the calculated dry weights are consistently higher, significantly higher, and much more consistent in the amended Duluth water. So this is suggesting that if we keep the quality of the diatom biofilms high by adding nutrients to the water, we can get much better growth over 14 days. And again, this is longer than an effluent test that we would do, but this is just a test of the principle. But adding these nutrients makes the mayflies grow much better. And just to kind of place it where these are in terms of this other plot that I showed. So again, the yellow dots were the lab water, the blue are the natural water. These red dots are these amended waters. And as you can see, it's quite good, quite comparable to natural water growth. And really the distribution was quite tight over those 4 tests. By the way, it didn't make a difference whether there was 3 or 1 organism per test chamber. They got about the same amount of growth. And the other thing we started doing in our diatom cultures is on day 7, after initiation of the diatom culture, we add the nutrients, the same amount of nutrients again to the dilution, to the culture water, the diatom culture water. And so this plot shows actual adult insects emerged, the live weight prior to releasing eggs of adults emerged from our cultures. And the light gray bars is just kind of the running average over time. And this is going back to mid 2017. And this isn't every single one of the insects that we do, but it's a random sampling. And as you can see, with the exception of in November of 2017, where for some reason we had a relatively high weights on a few individuals there. Starting at the red line, the first red line at November 1, 2018, that's when we started adding the extra dose of nutrients to our diatom cultures. And as you can see, the weights consistently went up. At the second red line is when we started culturing the mayflies in the amended water. And there really wasn't much of an additional boost there, but that was still probably not enough data to tell. So this is still a project that's ongoing, and we're still hoping to figure out more about this, but this is promising in terms of producing larger mayflies. So just to conclude, before I pass things on to Ning, in terms of general methodology, we're able to successfully execute daily changeovers with these tests that are started with less than 24-hour-old organisms. That was something that we weren't sure we were going to be able to do. We thought the handling stress might be too much, but it worked out quite well. In terms of organism age and sensitivity, the chronic values in these 7-day tests with less than 24-hour-old organisms were about 4-fold more sensitive than those we obtained using the 7-day-old organisms. Survival was never the most sensitive endpoint in these tests. Either calculated dry weight or biomass was. 7-day tests compared to the longer-term tests, they're quite similar in sensitivity compared to the 14-day and full-life chronic tests with sodium chloride. All these tests were done with sodium chloride. And in terms of food quality, in these control-only tests, adding these nutrients and silica to the reconstituted medium significantly increased the growth over 14 days. The real question is whether or not this has an impact on sensitivity of these organisms to effluents, and we've just started doing that. We've done one pair of acute tests, 96-hour acute tests in the standard reconstituted water and then the amended reconstituted water. The LC50s in the two are shown there, 957 for the unamended and 944 milligrams per liter for the amended. So far, no difference in sensitivity, but that's more testing needs to be done there. Again, next steps are repeat some of these experiments with mock effluents, test some other different types of contaminants, and then do further work to look at these amended reconstituted waters to really try to dial in the best growth and culture health for these organisms. And with that, I'd like to acknowledge the funding sources. This is funded by EPA, passed through USGS Columbia Environmental Research Center. And then I'll pass it on to Ning Wang, who's at the U.S. Geological Survey's Columbia Environmental Research Center.
Okay, everyone see my slides? Thank you for your interest. Please excuse me for my still very poor English, and I try my best. So as Teresa mentioned before, and the mussel is quite sensitive to many chemicals. And here is two examples of species sensitivity distribution for nickel and potassium, and the mussel and different from different family and tribes with the color symbols. Among the most sensitive species for nickel and potassium, and I specifically show the Ceriodaphnia, dubia, and Pimephales, the two standard species for the wet test. And the good news is the Ceriodaphnia is very sensitive, the number one most sensitive species for the nickel, but the Pimephales is not sensitive, and for the potassium, the mussel is much more sensitive than the two standard species. So in 2005, the ASTM already published the standard method for acute and chronic study with mussel, and for example, for the larvae Glaucidia 24-hour test with the endpoint viability, and for the chronic study and focus on survival and growth for 28 days. But there is no 7-day effluent test method and focus on survival and growth. And several years ago, when we worked with Fish and Wildlife Service in North Carolina and Oklahoma, we test some mock effluents or field collect effluents side by side with two standard species. You can see, and the mussel growth was affected at a lower concentration, and in mock effluent study and the field effluent study. So the two standard species do not present the sensitivity of mussel in these two cases. So in the past 2 years, we work with EPA Region 5 and the EPA Duluth to refine the 7-day test method with the common test species, which is mussel. fed market. And first of all, we'll try to find the best feeding rate for survival and growth. And then we also like to see what age of juvenile study we can start. You know, when we do a standard effluent test with Ceriodaphnia, the requirement is you have to use less than 24-hour-old Ceriodaphnia or less than 48-hour fat amino to start testing. But for the mussel, it's hard. It's not right now. It's not routine culture in many facilities. So it really depends on the situation. You collect adult mussel from the field and bring to the lab. You have to host the fish and the mussel culture and to transform from Glaucidia to juvenile stage, and you need 2 or 3 weeks, depending on temperature and species, and also shipping, acclimation, because for the test lab, we usually do not have the routine culture. So that's what we're talking about. It's hard to arrange the timing to start, and also when you have effluents from the field and when they come in. So it's likely you have mussel and available for a few days versus weeks. Is that change the sensitivity of the mussel when you start? So that's something we like to find out. And of course, then when we have the protocol, we like to do a round-robin study and to see whether other labs can do what we did here. So Teresa already mentioned the fed market is good species because they distribute across the United States and Canada. And the fed market is a long-term brooder. That means the mussel, the different species, and some is a short-term brooder, some is a long-term brooder. The short-term brooder, that means the Glaucidia mature very quickly, and so the adult mussel will release the Glaucidia in the water within a few weeks. But the fed market is a long-term brooder. The Glaucidia get matured in the female body and in the later fall, and the female can hold the matured Glaucidia over winter and waiting for the spring, the temperature raise. So what we can do is collect the adult mussel in the later fall or early spring and bring to the lab and put it in the low temperature water, so you can hold them for a long time and almost through the year, and you can collect Glaucidia for the culture or testing. So, and also fed market is relatively easy to culture in the lab compared to other species. So another good point is to use fed market is the fed market is a good surrogate to represent the sensitivity of other mussels. Here, there's a recent publication with did multiple testing with different chemicals, metals and ammonia, organic chemicals and major ions, with different mussels and from different family and tribes, and compare their sensitivity versus fed market. You can see here the EC50, more than 70% of EC50 from these 4 species is within the factor of 2 of fed market EC50. So that's the mussel really like a mussel, and so it's better way that you can see also the mussel sensitivity among different chemicals is quite similar, not like other taxa like fish or insects. So the difference could be substantial difference among the species. So to do the feeding study. So we did 7-day study, and with starting with 3 different age of mussel, and feed them 4 different amounts: 1 ml, 2 ml, 3 ml, 4 ml algal mixture. And the right side, you see that this is a commercially available algae, and it's quite easy to prepare the food and keep in the refrigerator and routine to use. And the end point is the mortality. So most of the time you see the empty shell, and very occasional time you see some swollen tissues. So we call them dead. And then also measure the length for the growth endpoint. To test the age sensitivity, we did a 7-day reference toxicant test of sodium chloride, and started with 3 age group of mussels, and used the sodium chloride 6 concentration, 50% dilution series, and we measured chloride and used chloride to calculate effect concentration. So this is a very simple. You can see all the material and stuff you need for the effluent study. Here is example the water bath holding the test chambers and 4 replicates and 6 concentration. And so for the daily renew, water renew, we use this kind of splitter unit to add additional water, and the splitter will divide the water in solution into the test chamber. This is a typical test chamber, about 300 ml chamber with a hole on the side covered with screen. So a very thin layer of sand on the bottom. So this is the results from the feeding study with different age of juvenile mussel and in 4 different feeding rates. So the dashed line is the average initial length at the beginning of the testing, and so you see the mussel length increased about 30 to 50% over the 7 days. There is no much difference among the 3 ages, and statistically analyzed, we do not see consistent results. For example, here for 1 month, 1 week old juvenile looks like 2 ml twice a day. They grow best, but for the 2 weeks old juveniles, twice 1 ml is the best. And when they're getting older, looks like even more, twice 3 ml is better, but it's not significant. Overall, the mussel grow substantially over the 7-day study, but we did notice there's quite a bit debris in the one feeding treatment is like feed them once large, 1 4 ml, and that is not a very good sign to have a lot of accumulated food in the beaker. So our recommendation is about 2 ml twice a day, and for when we start testing with 1 or 2 week old mussel, and when we start with 3 week old mussel, we should feed them a little bit more, 3 ml twice a day. So with this feeding rate, we start our sodium chloride testing with the 3 different age of juveniles, and this is control survival and control growth here, and they grow like we found in the feeding study and grow a lot, more than 40%, and survival is 100% for the 3 study. And the mussel and the younger mussel, the 1-week-old mussel, the survival was affected at a high concentration, but not see this survival effect when they're 2 or 3 weeks old. But we see the more sensitive endpoint for the length and the growth effect at a much lower concentration. And if you compare these 3 age difference, the length endpoint is not much different. So when we have this method, we ask around at the meeting or email and see whether any other lab like to do a round-robin study. Informally, we ask the people, and there's 3 university lab and 5 private sector lab and 5 government labs like to join us. And some lab have some mussel testing experience, and some not. And so one lab even have now toxicity test experience, but they know how to culture the mussel well. So just informally to group these people to do the round-robin study. So this is the summary of test condition we send out, and I just want to emphasize we use the EPA moderately hard water, and we also use the same batch of juvenile mussel culture at one Missouri State University and sent to all the lab at the same time, and to do the sodium chloride study. And at the end of test, we checked survival length, and we also use the dry weight based on the length and weight relationship, like Dave just mentioned before for the mayfly, and the mussel is very small, like a 0.2, 0.3 millimeter at the beginning, and so it's hard to measure the dry weight each time. So the reason to use dry weight is better to calculate EC20, and Dave already mentioned. So we use the equation we had before, and just want to emphasize here, and the slope is 2.71, and it's almost identical as Dave mentioned for mayfly. So it's close to 3. That's quite normal. And this is an example from one participant lab, and based on length and dry weight, you can see based on the dry weight, the dose response is much better, and the value is quite different between the length and weight data. So this is the results about after the 7-day test from the 13 labs, and most labs face a big challenge to recover the mussel from the sand. So the reason we made a mistake, we didn't save the sand less than 0.2 millimeter, which is smaller than the mussel. So we send out the sand particle size is quite big than we originally planned, and make it difficult to recover the mussel. Another thing is we didn't explain very well during our conference call before the study how we are going to recover the mussel, and there's some kind of technique. It's very helpful. So when you gently swarm the beakers, then you can concentrate the mussel on the center of the beaker and on the surface of sand. So you can quite easily to cover that way. But anyway, the mussel lab gets very good recovery, and except number 4 and number 11 lab, they have like 80 or 85% recovery. The survival based on recovered mussel is all the lab meet the accepted criteria, more than 80% control survival. So this is the example and how people use the digitized software to measure the length and get the length value. And so here is the length increase among different labs. We see the large variation in growth among the labs. However, in most cases, the length increase about 20%. So we're going to explore more why there's much difference in between several labs. And for the response to the sodium chloride and the survival, we only 2 labs see some survival effect, number 6 and 12. But we see the growth effect. All the labs see the growth effect, and from 280 to about 1,300 milligram chloride and affect the growth. So the growth endpoint is more sensitive again. So this is the EC20 among different labs. So the difference was within a factor of 4.5. We see there's a large confidence interval in several labs, and mainly is because only the highest concentration have some substantially reduce the growth, and the other not. So that's why there's some high variation over there. Here I add one green square point is the sodium chloride test results tested in our lab, and I mentioned before, and you see within the lab variation is similar to the inter-lab variability among the 13 or 12 group. I should say 12 labs right now here. So if we have to compare the inter-lab variability to other studies, and there's a 4-day fed market acute study based on survival. The CV is equal 42, is almost identical, and then this one, and although EPA more formally organized round-robin study with Ceriodaphnia and Pimephales, it's 35 and 17. But again, this is not an apple-to-apple comparison, and there's different number and organism and test source different. So just give you some idea. So the conclusion, so all the group of aging mussel and grow substantially over 7 days. So the accepted growth measurement can be determined. So also the different mussel, different age of mussel have the almost same sensitivity to sodium chloride. So the effluent test can be start with 1 to 3 weeks old fed market. So that's give us some flexible time to start testing with the effluent. And finally, all the labs meet the 80% control survival criteria, so the variability is quite low. So the methods have acceptable precision and can be performed routinely. So with that, so for the future study, maybe Teresa can make some points for the future plan.
All right. This is Teresa. I don't have a slide that covers this, but I wanted to talk about an overview that we are not done with this study. We are hopefully going to be able to do some comparative testing with the mayfly, the mussel, the Ceriodaphnia, and even maybe the fathead minnow with a sample from effluents, or we may have to be able to create some samples to test, but that's our next step. And I think the important part is that we've made some really good progress. We're getting closer to being able to have a protocol that would be a standardized protocol, so that if everyone was going to start testing these, they would be able to do it the same, as close as we can, to the same way as we do for the other species. And with that, I'd really like to conclude that we appreciate your time and attention to us on our seminar.